possible que ça soit de la fibrose, non, ce problème. Est-ce que je peux voir? Ah, c'est bon, t'as caché, je les os. Ça destroy under the skin.
We are interested in the basic mechanisms important for the development of lymphatic vessels. Blood vessels bring oxygen and nutrients to tissues. Lymphatic vasculature collects liquid, proteins, and immune cells that escape from blood vessels and transport them to the lymph nodes, which is very important for the immune response. And finally, to the venous circulation, which is essential to maintain fluid balance in the organism. Lymphatic vasculature contains two functional compartments: capillaries and collecting vessels. Lymphatic capillaries communicate directly with the interstitium, and they are responsible for the uptake of substances and cells. The role of collecting vessels is the transport of lymph to lymph nodes. Efficient lymph transport depends on correct functioning of both compartments. Since few years, we have been working on the mechanisms of specialization of lymphatic vessels. Simply speaking, we want to understand why and how lymphatic vessels decide to become capillaries or collecting vessels. In this paper, we focused on the morphogenesis of one of the most fascinating structures of collecting lymphatic vessels, lymphatic valve.
Lymphatic valves are important to prevent lymph backflow and to ensure efficient lymph draining. In this study, we focused on the early stages of lymphatic valve formation and identified 4 steps. The process starts with the upregulation of 2 transcription factors, PROX1 and FOXY2, in a subset of cells. These cells segregate into a cluster on one side of the vessel. Then they assemble into ring-like territory, condense, and fold into the lumen to form the valve leaflets. Using genetic models, we studied the mechanisms of early stages of lymphatic valve morphogenesis. We found that the assembly of a ring-like structure requires the gap junction protein connexin 37, which acts upstream of calcineurin and FAT-C1 signaling. In turn, calcineurin and FAT-C1 induces condensation of early lymphatic valve site. We next asked what is the possible trigger of lymphatic valve development, upstream of connexin 37 and calcineurin. Lymphatic valves are frequently formed at vessel branching points. As we know from hydrodynamics, fluid flow is disturbed in such regions. We visualized lymph flow in embryos, and we observed that before lymphatic valves form, some regions of the developing collecting vessels are subjected to reversing flow.
We hypothesized that the disturbed flow in some regions of developing lymphatic vessels could be a trigger of lymphatic valve formation. To test this idea, we studied responses of lymphatic endothelial cells to different types of shear stress in vitro. Oscillatory flow induced connexin 37, and this process was dependent on the presence of PROX1 and FOXY2. These transcription factors were also required for the calcineurin activation by oscillatory flow. In turn, calcineurin activation required connexin 37. Finally, oscillatory shear stress specifically induced FOXY2. One of the most interesting observations in our study is the fact that PROX1 and FOXY2 control general responses of lymphatic endothelial cells to shear stress, such as cell elongation or stability of cell-cell junctions. We propose a model of lymphatic valve development where the disturbed lymph flow leads to upregulation of FOXY2, connexin 37, and FAT-C1 activation. Together with PROX1 and mechanical stimulation, this is translated into a discrete cell signaling pattern and morphogenetic event, lymphatic valve formation.
Of course, there are still many open questions, some of which we think are fundamental for vascular biology in general. How do lymphatic endothelial cells sense flow? What is the molecular basis for cooperation of PROX1 and FOXY2 with mechanotransduction? How does endothelial cell identity, arterial, venous, or lymphatic, define responses of the vessels to mechanical forces?